Okay, okay. Yes, sir.
Okay, so then I continue. So regarding the city, as I said, in our institute, we are working a lot on the development of polymeric materials. We are more than 10 research groups that are working on different aspects of the development of polymeric materials. And one of the aspects that we're exploring is the use of polymeric materials for biomedical applications. I'm leading one of these areas. Yeah, so we are 2-3 groups that are already established in the institute, and we have now new junior groups that are leading research groups. And the idea is that over the years, we make this area a little bit stronger in Polymat. And also within the environment that we have here in San-Sebastain. Just to give you an idea, you have a look here on the SES Nano. There was a publication last year where there was an editorial highlighting the activities that are taking part here in San-Sebastain, which is being called a city of nanoscience and technology. This is a very nice publication. You can have a brief summary on all the activities that are taking place here in the city. And it's a nice sentence. They said the San-Sebastain nanoscience community has contributed considerably to SES Nano with more than 100 publications during the past decade. So you can see it's a very, very active community. And of course, here we always try to get also very prominent visitors. And last year we had a visitor that you know. So I'm just trying to move to the next slide. Here you see we have Professor Jayant Khandare visiting us last year. He was for a very short visit here, and we were of course honored to have him talking in our institute on November, and he was talking about the design and the clinical evaluation of biopolymeric translation from lab to clinic. It was a very interesting talk where he took us through a journey on this approval on the CTC technology in OncoDiscover. But well, just to give you an overview about the topic that we will discuss today, we'll be talking a bit about the motivation of our group. I will give a brief introduction about polymer therapeutics and responsive nanogels, and I will give also a bit of introduction about the use of these responsive nanogels for topical drug delivery. And I will just try to summarize 2 of the examples from our group where we explore protease-sensitive nanogels and thermoresponsive nanogels. Here you can see a picture of our research group, the responsive polymeric therapeutic group. We have several members. And as I said, we're still growing as we are getting junior group leaders that are joining our group, and they're also leading their own activities. And in this case here on the picture, we have Annabelle Loki, that she joined our group in February with her own group. And she will be there at least for another 5 years. So in our group, we're very active in Twitter, so you can follow us as Calderon Lab. There you will see the latest news of our group. And we also established a YouTube channel where we are now uploading some of the talks that we recently hosted and in virtual symposium. As you're having these webinars, we had as well a virtual symposium with speakers from all around the world that show the developments on nanogels for different applications. So there you will see I will be posting once a week the talks that we received during this presentation.
Oh, no.
So regarding the motivations of our group, the very general motivation is trying to explore the different physicochemical features of responsive materials to enable the crossing of biological barriers. Yeah, we recently discussed this in a review we wrote in Journal Control Release, where we were focused on the development of responsive nanogels. But in our group, we are also thinking on beyond responsive nanogels, also in other polymeric architectures. And the idea is that we try to explore how the different chemical modalities will influence on the properties of the materials and how these properties of the materials could then be useful to cross biological barriers. Yeah, we all know that crossing biological barriers is very important when we want to deliver a therapeutic or a diagnostic action behind these barriers. And here we rely on the engineering of the nanomaterials to have a certain response and to have a stimuli responsive material to release a drug or to release a diagnostic agent, etc. So regarding this motivation, a bit more in detail, in the group we have 4 big areas of research. We focus a lot on the use of dendritic polymer for diagnostics, and this is an activity that we started developing together with Jayant in Berlin when he came to visit us, and we joined the same group as postdocs in the year 2008, and we started working with dendritic polyglycerols. There we started conjugating drugs to these polymers, and we use them on different applications. And later on in my group, we also explored a lot on theragnostics. We as well explore the use of these dendritic molecules as building blocks to prepare thermoresponsive nanogels that were later used for topical drug delivery. And these will be the two topics that I will discuss today a bit more on detail. But in any case, I will also like to say that we work in different approaches to provide smartness properties to the polymetic materials, for instance, to deliver and to release a cargo upon changes on the endolysosomal pH, for instance, or as well to play with light, with near-infrared light, to induce a conversion of this near-infrared light into heat, and thereby use this heat to change the properties of the materials and also to kill cancer cells. Somebody said that is not able to listen to hear my voice. Can everybody else hear my voice? I just got this message on the chat.
We can hear, Marcelo. You continue. We can hear.
Then I continue. And as I said, today I will focus on the 2 upper corners of this motivational research experience slide. I will be starting now with the dendritic polymers for the part. I have an echo. We need somebody there to mute their microphone, please. Namrata apparently has it on. So regarding polymer therapeutics, we all know here, or I will assume that most of the people here in this audience is familiar with these concepts where we try to use conventional chemotherapeutic agents that have a lot of limitations on their use because they are small molecules, have a low solubility in water, they are widespread in the body, and they have a fast clearance, and they have a low selectivity to target cells, thereby having a narrow therapeutic index. And what we try to do with polymer therapeutics is combining these small molecules with bigger particles or bigger polymers in a way of getting better properties, enhancing the solubility, reducing the body clearance, enhancing the residence time in the body, improving their targeting to the site of action, and controlling, of course, the release profile of these materials. And at the end, we try to improve the therapeutic index of them. So the very common concept that I will believe you're familiar with this is enhanced permeation and retention effect, the EPR effect, by which smaller molecules will penetrate both in normal tissue and as well as in disordered tissue. Typically we are talking about here solid tumors. But in the case that we have macromolecules or molecules with a certain size, this will not be able to penetrate on this normal tissue because the tissue is very well packed, but they will be able to penetrate on the disordered barrier tissues. For instance, on the solid tumors where these barriers are very leaky, and macromolecules will be able to penetrate there, and because of having of a non-working lymphatic system, they will also be able to be retained there for longer times. So once these materials are retained there, we can make use of different biological clues. As I'm a chemist, I like to say these are chemical clues. One of the chemical clues we can very clearly see here is a decrease on the pH from physiological pH of 7.4. We get into lower pH of 6.5 approximately on the surrounding of the tumor cells, so-called tumor stroma. And then we also have an enhanced activity of certain cells, of certain enzymes like matrix metalloproteases, MMPs. They will also have an overexpression of certain kinds of tumors. And when we get closer to the cancer cells, our nanoparticles could go inside of the cells by different mechanisms, most of them endocytic mechanisms. And whenever the particles get inside of the cells, they will also see another drop on the pH when they move to endosomes of different stages. pH drops approximately 1 unit, and this could drop even further when it goes inside of the lysosomes, which are a later stage of the endosomes. And then here we'll also have an overexpression of enzymes. Typically, enzymes that we can find here are catepsins. One of very popular one that is used to cleave linkages between polymers and drugs are catepsin B, for instance. And if our molecules are able to escape these endosomes or lysosomes, they can diffuse into the cytosol of the cells, where we could see a reductive environment that could also be used to break linkages between polymers and the drugs. So this will be some of the clues, and this is how we can use polymer therapeutics in a way of improving properties of the conventional chemotherapeutics. And a very common example that is already in the market and is used in clinics for several years is the so-called Doxil. Here we have a liposomal formulation of doxorubicin. Doxorubicin is a cytostatic molecule that per se has several issues. One of the issues here, Supriya wants to say something. You have a question? He raised his hand, I think. Okay, no. So, well, doxorubicin has one of the issues. It has cardiotoxicity. Yeah. So, beside others, of course, no. So, and a low specificity, fast clearance, as we can see here on the residence time of doxorubicin after being injected in a patient, it clears very, very quickly. So here we have concentration in blood, doxorubicin concentration in 24 hours. But at the moment, this molecule is encapsulated within this liposomal formulation. The toxicity decreased a lot, and the residence time also increased. Yeah, so this is a very interesting system. This is a very nice profile, at least for the first injections of Doxil. But of course, we have issues with this sort of materials where we only encapsulate drugs inside of a liposome, where we can have an unspecific leakage of the drug during circulation. And then this is one of the issues that current science is trying to overcome. In the direction of overcoming these issues, people have been modifying chemically doxorubicin to convert it in a prodrug. So here we have the structure of doxorubicin. I hope you can see here my pointer. And then doxorubicin was here modified on the ketyl group to be coupled to a linker. And this linker has a nitrogen bond that is stable at pH 7 or physiological pH, but is degrading at acidic pH. Yes, so this will be an interesting product when it comes to accumulation on tumor over the pH decreases. And we have a very interesting functional group here, the malimido group, that reacts very, very quickly with thiol groups at pH 7, between 7 and 8. And within 5 to 10 minutes, this reacts. It's basically a click reaction here. It's a Michael reaction. And in the case that this molecule is being nowadays explored in clinical trials, what happened with the drug is after the injection in the blood, this gets conjugated, chemically conjugated now, to the thiols on the albumin that we have circulating in our blood. And by this conjugation with albumin, we have a very safe system that is not active, or at least decreased activity and toxicity, while still is coupled to the protein. But at the moment, this molecule gets into an acidic environment, like in the tumor. This gets cleaved, it's been released, and then we have an activity against only the cancer cells, or at least specifically against these cancer cells, and not so much against the normal tissue. So this is now in clinical trial 3 in several countries in Europe and in the US, and also in different indications. But you can see here that as we have only one active thiol in albumin, this loading capacity of this approach is relatively low. So one of the approaches, of course, is to replace this biopolymer by a dendritic polymer. And this is what we started doing together with Jayant when we joined the group of Rainer Hack in 2008. And we could couple multiple copies of doxorubicin into the dendritic polyglycine. I will not get too much into details today about that. We have several publications in that field, and with Jayant we published more than 10 papers in that field. But if you're interested, you can, of course, go into details of the publications or contact me and I will send you them. But today I also wanted to raise a question, and the question here is whether EPR effect is enough. Yes, so we know that still, even though we have this interesting permeability and retention by the molecules, there have been several studies where they show that the big particles could very well penetrate on tumors, accumulate on tumors, but they do not penetrate too deep. And this, of course, will have a consequence that not all cancer cells will be stopped on growing because of on these therapies, whereas smaller molecules, a little bit lower than 50 nanometers, could penetrate a bit deeper. Yeah. So then what we propose here is that we can have nanogels or nanoparticles that it will be able to penetrate with higher sizes, like the large nanocarrier represented here. But once they get accumulated into the tumor, they are degraded and they can penetrate deeper. And this is just a simplified, of course, a very simplified model of the EPR effect and the tumor tissue. And we have to consider, of course, that there are many other types of cells beside the cancer cells in the tumor that we are planning to also consider in the future, because more than 50% of the tumor mass is typically comprised of different cells, like for instance macrophages on different stages, fibroblasts, and so on. But well, just going into the direction, yeah, so I will try to talk a bit closer to the microphone. Going into the direction of these degradable nanoparticles, we have different approaches. One of them is using biomacromolecules as nanotemplates. So we prepare micelles of casein, in this case, casein, just a protein typically found in milk. They form typically by themselves micelles, and here we cross-link them through different kind of linkages and a collaboration with a group of Argentina. Dr. Matias Pique worked a lot into this area, and we demonstrated that we could control the degradability and the specificity of the system toward proteins that are overexpressed on the tumors. Another approach that we follow is using polypeptides. Yes, so we had here a collaboration with Professor Maria Vicente from the Institute Príncipe Felipe in Valencia, here in Spain. And we synthesize nanogels where we cross-link different polypeptides. In this case, we were using polyglutamic acid that is used a lot on clinical trials for drug conjugation. And here we cross-link these materials through click chemistry and form nanogels with control sizes, control functionalities. And then by encapsulating doxorubicin within these materials, we were able to demonstrate an antimetastatic activity of these nanogels when they were encapsulated with doxorubicin. But the topic that I will explore today more in detail is developed by Gregor Nagel. He was a PhD student in my group. He graduated last year, and he prepared degradable nanogels on a more engineered design. So here what we propose, as I said before, is a material that in the case of the solid tumors will be able to get accumulated on the solid tumors because of the big sizes. And then by degradation, they should be able to penetrate deeper in the tumor and then release the drug. So we have here a multi-stage degradable and release system. So we have a nanogel that it will respond to activity of MMPs. So these proteins that are overexpressed, enzymes that are overexpressed in the extracellularly and different type of tumors. And upon this cleavage, we will get degradation of the material. And here we included as well a FRET system that it will also serve as a diagnostic approach. Yeah, we will recover fluorescence and we will see fluorescence of the material upon degradation. And then once this gets small, if the smaller fragments get deeper into the tumor cells and they penetrate and face smaller pH than 7, they will also be able to release a drug that it was chemically conjugated to these fragments. So following the concept of conjugation of doxorubicin we showed you before. Gregor did an excellent job, and he followed some of the techniques that were developed in the group of Rainer Hack, where we chemically functionalize polyglycerol with cyclooctane groups, and then we use a system that it was published already in literature that it was known to be degraded by MMPs. Here we have a certain sequence, peptide sequence. Here we have the FRET pair, and then at the end, on the terminal groups of this peptide, we conjugated polyethylene glycol that it was readily functionalized for click chemistry versus the cross-linker of polyglycerol. So we did this an inverse nanoprecipitation that is a very mild, facile, scalable technique. Yeah, we could control these run reactions even in water, and then we could control, as you will see now, the properties of the nanogels. So we could fine-tune the different sizes of the nanogels, ranging from 200 to 700 nanometers. We could also play with the peptide cross-linker density and also on the functionality of the polyglycerol cross-linker. We got a spherical looking particles with sizes, as I said, very well controllable. And what it was also very interesting that we could see that upon controlling the incorporation of the peptide cross-linker and the functionality of the dendritic polyglycerol cross-linker, we could as well control the different degradation kinetics, as you can see here. Yeah. So here we plot the recovery of the fluorescence. So as we propose to have a nanogel that will be quenched by the FRET effect, at the moment that this is degraded upon activity of MMPs, will recover fluorescence, and this could serve as an indication of degradation. And this is what we can see here. No? So the recovery of fluorescence upon incubation with MMPs is very quick. You see, depending on the functionalities of the dendritic polyglycerol on the peptides, this could get a little bit slower. This is what we can see here. We can slower the degradation by this control. And what we also can see is that the size at the very beginning, when we have a non-degradable material, stay almost the same. So perhaps we have a bit of aggregation or deposition of proteins on the surface of the nanogel. But when we have a degradable nanogel, what we have here is that the size increases a bit at the very beginning, and this is very common when a material is degrading, like nanogels enhance the size because they start aggregating, they also start opening the structure. But then a moment when the fragments start to come out, we can see that the sizes decrease up after six hours approximately. We can see that the smaller fragments now are coming out of this nanogel structure. So the question now, the more interesting question is whether we can also see this on a closer scenario to reality. And for this, Gregor developed some multicellular tumor spheroids in our group that were a very good model for the penetration of solid tumors in vivo. Yeah. And here what we did is we did virtual sections with a confocal microscope, but we also did actual section in by histology afterwards. And what we could see here when we have these multicellular spheroids using the same cancer cell line than in the former case, I think it was HeLa, and here we have a co-culture with fibroblast. We could see that whenever we use a free dye as a control, we could see a deeper penetration of the dye into the tumor. Yeah. In the case we have a non-degradable control nanogel, we can see here when we compare the intensities that the penetration is hampered. But in the case we have a degradable nanogel, the penetration of this small dye that it was conjugated to our nanogel is deeper than a non-degradable material. Yeah, so we can see here that the nanogel is penetrating. Here we have the green line here, the degradable nanogel is penetrating up to 60 micrometers in this level, where in the case of non-degradable nanogel we have almost no penetration. You can see almost no penetration at all. This was only the label nanogels, but then of course would be more interesting to see what happened when we couple doxorubicin through a pH cleavable linker to these nanogels. And here we follow doxorubicin fluorescent. This is the free control where we have the free drag. We can see that penetrate very deep onto the solid tumors. In the case we have the non-degradable control, we have a lower penetration. But then when we have our degradable material, we have a much deeper penetration of doxorubicin. So here, doxorubicin will not only account for the penetration of the fragments of the nanogels, but will also account by the penetration of the drug itself. Yeah, and this is what we can see here. Whenever we have a non-degradable nanogel, you can barely see penetration of doxorubicin, but in the case we have a degradable nanogel, you have a strong penetration of doxorubicin, and you also have a co-localization with the nucleus, which is the target site of doxorubicin in most of the cases. Of course, we also assay the activity of these materials, and we could see that whenever we have a nanogel without doxorubicin, there's almost no toxicity. In the case we have doxorubicin with a pH cleavable linker coupled to a non-degradable nanogel, there is almost the same. We found the same, almost the same cellular proliferation. In the case we have free doxorubicin, of course, this decreases the proliferation a lot. But in the case we have our degradable nanogel, we have a very good activity of doxorubicin in these cases on these tumor spheroids, which is a very nice proof that this material have a very strong potential on this multi degradation and release profile. This material was published in Theragnostic, and this was published this year, and we made it to the cover, and you can see here we have a nice representation of what we tried to do. You know, long circulating nanogels into the blood vessels, but at the moment they get into the tumor sites, they should be able to degrade into smaller fragments. They will shine on, and then upon shining on, they will also penetrate deeper and also reach the nucleus of different cells. So with this, I come to a conclusion to the first section of my talk. The conclusions were already discussed right now, but of course the perspective are still there, and we are planning now to continue this research with funding from the Ministry of Science here in Spain, where we want to explore how these materials could be also used to target certain cell populations on the tumors. And of course, we would like to target macrophages, change them from the different subtypes to the anti-proliferative subtypes in the tumor. And of course, then we will also explore how these interact with fibroblasts, so on and so forth. So this will be the first section. So now a second section, another 10 minutes of presentation. I would like to talk about the development of responsive nanogels for topical drug delivery. Of course, I will start with a brief introduction to that, but before that, I would like to say that this was done in all the results I will present today was done in the frame of the Collaborative Research Center in Berlin, in the Freie Universität Berlin before I moved to Spain. And here we were part of a big network, approximately 15 groups that were working on different research areas. I was belonging to research area B, sorry, A, working with synthesis, drug uptake, and labeling of the materials. So this is the blue part here on this sphere. But then there were other groups responsible for physicochemical characterization, also for the testing on relevant models. Yeah, so here we have dermatologists, we have pharmacologists working, testing this material on 4 different models, in vivo and in vitro. And also we have a central system or a central group that it was taking care of the toxicological assessment of the materials. So regarding the concepts here, what we try to propose is the use of our nanocarriers to enhance the penetration of therapeutics into the skin. So most of the drugs are being blocked by the different layers of the skin, and they cannot penetrate because of the size they have, because of the aerophilicity balance they have, and many other properties. Some of them can make it, and they can make it through intracellular mechanisms, intercellular mechanisms. This is the more common one for small molecules with molecular weights lower than 500 Dalton. And then there is also a very interesting one, molecular, that this is very promising for the application of nanomaterials. So just as an introduction also, I would like to say that the skin has different layers. The one that is very important for this talk today is the so-called here stratum corneum. And this stratum corneum is basically a very packed layer of dead cells, and they're responsible for mostly for the filtering properties of our skin. And then we have, of course, viable epidermis, which are together, they make the sections of epidermis. The viable epidermis have live cells, and then we have also dermis underlying the epidermis. And here we will find many other kind of cells. We have blood vessels and so on. So the fields of applications are several, of course, in the area of cosmetics, anti-aging, for instance, also in the treatment of acne. But there are many other common diseases like psoriasis and atopic dermatitis where we could have a field of application. Very interesting but rare diseases are also a series of diseases called autosomal recessive congenital ichthyosis. And in this disease, there are issues or congenital issues by which the patients are not able to produce certain proteins that are very important for the packing of the stratum corneum, for instance. And these proteins, of course, are very, very difficult to penetrate on the skin if we would like to administer them from outside. So in our case, we will also propose the use of our nanocarriers toward this application. So nowadays, there are different fields on how one could achieve the penetration of molecules inside of the skin. Some of them using penetration enhancers, either by physical disruption of the skin or chemical disruption of the skin. And there are others that are using, I'm having issues with my next slide. It's not coming. Yeah, here it comes. And nanoparticulate systems that could work as reservoirs. And the typical examples that are nowadays being used are nanocrystals. And these are basically the crystals on a nanometer size of the same drugs. And then there are also options to use polymeric nanoparticles. Polyesters are used a lot in this area, for instance, and what are very fashionable nowadays are the solid lipid nanoparticles because they're very, very flexible, they're able to penetrate, and they have the right aerophobicity to penetrate into the skin. So just thinking on these nanoparticulate systems that are being used nowadays, our nanogels could have a property somehow in between polymeric nanoparticles and solid lipid nanoparticles because we have the character of polymeric materials and the functionalities they have and the stability they have. And the other side that we also have from the solid lipid nanoparticles are their strong flexibility and deformability. And to a certain extent, also the possibility to have a proper hydrophobicity or tuning of the hydrophobicity that we would like to have. So in our research collaborative center, we propose to synthesize these materials at tridimensional networks and to study different properties of the materials. Yeah, so we explore at least all these fields on many different aspects. So that this was research that it was performed over the period of 5 to 6 years. So we have several publications in the field. So if you have questions about this, I could also send you publications regarding the field. And today I will only focus on one very small aspect regarding the mechanisms of penetration of the materials and delivery of proteins. But what we propose here is to play with the thermal gradient that the skin has per se. Yeah, so we have the very outer surface of the skin at temperature of 32 degrees. And when this increase a bit to 37 is when the nanoparticles penetrate into the stratum corneum. So this we will have here a gradient of temperature of approximately 5 degrees that will be used to change the properties of our materials. If we have a thermoresponsive nanogel that is fully swollen at 32 degrees, and upon penetration into the first layers of the skin, will face an increase of temperature that it will induce a shrinkage and a release of molecules that were encapsulated within the structure, either water molecules or drug molecules. The other option, of course, is to use an IR lamp and increase the temperature artificially from outside. So from the exterior, we could reach temperature to 40 degrees without having major harms on the skin cells. And this is fully acceptable. And of course, the other option that we have is to play with the biodegradation that it could be exerted by glutathione, for instance, that is predominantly found, for instance, on mucous. But we also have certain proteases like catepsins, for sure, in the skin that it could also be used to induce degradation of the nanomaterials. And we revised this concept in a review recently that it was written by Neha Tiwari in our group, and she's actually from Pune, and she very well presented the different properties or possibilities that our nanogels will have on this regard. So a bit more on details on the synthesis. What we use here is again a dendritic crosslinker. This is represented by these gray spheres, and these gray crosslinkers were connected with each other by thermoresponsive polymers, which are these lines here. And these thermoresponsive polymers, upon a change on the temperature, will shrink, will expel water, and they will also expel cargo molecules, yeah, either dyes, drugs, etc. So here we say that there is no audio. Can you hear me?
I think there are some exceptions. Maybe their network might not be good. So we can hear. Go ahead.
Okay. Just in case there is no audio, please write me, Jayant.
Yes.
So this will be the general model. So we will have a molecule that is fully swollen at a certain temperature with a lot of water inside, a lot of cargo molecules. And then when we increase the temperature, in the case we use a thermoresponsive polymer of the type of LCST, the molecule will shrink, will still be stable. Yeah, this will still remain in solution or in suspension, whatever it is, but then we will release a cargo molecule. So a bit more on chemistry. I will not get too much into details with the chemistry, but I would like to anyway show you the building blocks we use in our group. As I said, the chemical crosslinker here, it was this dendritic molecule, polyglycerol, very easy to functionalize, very biocompatible, and the thermoresponsive polymers were compressed as different thermoresponsive monomers or polymers that are commonly used. Now, the one that is very, very famous is the N-isopropyl-acrylamide, so-called NIPAM, and this NIPAM has a very good transition temperature of around 33 to 34 degrees, which pretty much fit into the hypothesis we have now here, playing with temperatures between 32 and 37 degrees. But then this could also be copolymerized with another monomers to either increase or decrease this transition temperature. And one common monomer that we can use is NIPMAM. Yeah, it's very similar to NIPAM, but with the methyl group here on the main chain. And this will enhance the transition temperature from 32 to 33 from NIPAM, actually to the one of NIPAM that is around 48. If we copolymerize these two materials, we can have the value we want in between. There are other options to use ethylene oxide methacrylates, which are also upon the different composition of the comonomers, will give possibilities to play with the transition temperatures between 25 to 80 degrees Celsius. But then there are other options, of course, like PBCl and also thermoresponsive polyglycerol units, what we call TPG, which actually is a polyether backbone thermoresponsive material. Again, here, when we combine different compositions of comonomers, methylated or ethylated, we can also modify the transition temperature of these polyglycidyl ethers. When we play here with the different properties and the different sizes and the different compositions, of course, we have to consider that these materials could be toxic, especially when we run radical polymerizations to prepare them, especially when we use surfactants that could be harmful to cells. So then we had to test their properties regarding tolerability. And for that, we tried different studies. Of course, the simplest is to test cell viability. But then we went a bit deeper to study whether they will have an irritation potential, whether they will generate oxidative stress, or whether they will have genotoxic effects. And in all cases, we didn't find any of these effects. So our materials were rather biocompatible, at least in the frame of this concentration we tested and in the frame of this test that we ran. Afterwards, we analyzed how the penetration mechanisms of the materials could work. Yeah, and we needed to know what happened with the nanogels once we put them in contact with the skin, and what happened with the cargos once the temperature changes. And here we did a collaboration with the Charité Hospital in Berlin, and this was led by Fiorenza Rancan, that is shown here on the top right corner. And right below, we have a picture of our student that was responsible for this project, Mikhail Gulbodarian. And he synthesized nanos with different compositions, yeah, with NIPAN, with oligoethylene glycols, or with polyglycerol ethers. And what we could see in all these studies, whenever we follow the nanogel, that it was fluorescently labeled here in red, or we encapsulated a molecule like fluorescein here in green, what we could see in most of the cases, and here is very, very clear, is that the nanogels actually sit on the stratum corneum and they don't go beyond the stratum corneum. When we check what happened with the cargo molecule, we can see the cargo molecule could penetrate very deep on the skin, and it could stay there on the skin. Yeah, so these are pictures after we washed these human skin models. And whenever we did quantification on this, we could very interestingly see that the polyether backbones based polymers were the ones more responsible for the high penetration of the cargo molecules, yeah, both in stratum corneum and epidermis and in dermis, yeah. And whenever we try to see what happened with the structure of the skin, we could very nicely see and that the molecules that are based on polyether backbones are changing the skin properties a lot. Yeah, so here we analyze together with Professor Ruhl, and he's working on the physics related department from our chemistry department in Berlin. And he analyzed by stimulated Raman spectrum microscopy how is the distribution of lipids and proteins on the skin. He did that following two peaks for lipids and proteins. He normalized the two peaks for the so-called normal conditions of the skin. This is the value here we have in control. But at the moment he started analyzing this ratio between these two peaks, he could see there was a strong disturbance on those systems where we have the polyether backbone nanogels, which were the more responsible for the high penetration of the cargo molecules. This, of course, has a strong correlations on the aspect of the skin. We can see that the skin is deorganized, and it has, into the visual aspect, a strong hydration, yeah, which was a hint for us to understand what could be the mechanisms of penetration of the cargo molecules inside of the skin. What I didn't mention at the beginning is among the chemical penetration enhancers, water is one of them that is very prominently used on different applications. Of course, hydration to the skin, as we probably most of us already know, enhance penetration of molecules inside because basically enhance the porosity of the stratum corneum. So with this in mind, we have another PhD student, Ernesto Osorio Blanco, that he's handing in his thesis on the next days. Also working with Fiorenza Rancan and the group of Professor Ruhl. Here we explore the use of not only nanogels, but also nanocapsules, because here we will have a material that it will be more flexible, and ideally will also have a strong or a higher content of water within it. And he synthesized these materials, he tested them regarding different properties on the mechanics, thermomechanic properties of the materials. We study how the mechanics of the materials change whenever we change the temperature of the materials above and below the transition temperature. And then we also study what happened whenever we put them in contact with the skin. In this case, we compare these two systems, but in one of them, we also have the nanocapsules still with this, the sacrificial core inside. So we have a material that is very, very flexible, which are the nanocapsules in this case. And then we have materials that are rather rigid whenever we have the silica nanoparticles still in the core that were used as sacrificial core templates. So this is a very heavy slide, but I will just summarize it by saying that what we analyze in these studies also by Raman spectrum microscopy is the penetration of deuterated water, D2O, D2O. And here what we analyze is how the materials are able to enhance the hydration of the skin. And this is what we can see here. So the D2O intensity. And whenever we use only D2O in contact with the skin, we can see, of course, a penetration of the D2O on the skin. Yeah. Whenever we put in contact with the skin the silica nanoparticle core nanocapsules, we can see that there is also penetration and a little bit higher on the viable epidermis as compared to only water in contact with skin. But at the moment we have our systems as nanocapsules, you can see that the water penetration enhances a lot comparatively to the water and the silica core nanoparticles, both in epidermis and in dermis. Sorry, stratum corneum and viable epidermis. This is what we are seeing now at the moment. And at the moment that we enhance the temperature by irradiation of an infrared lamp, we can see that we enhance the concentrations or the amount of water that penetrates into the skin, both for the stratum corneum and the viable epidermis. And this, of course, has a correlation as well with the cargo molecule penetration, but I will not get much into the details with that area, just not because of the matter of time. But another aspect that we explore here, beside the penetration of cargo molecules and water inside of the skin, is penetration of the nanoparticles into the hair follicles. As I said at the very beginning, hair follicles are a very interesting target for nanoparticles because of the properties of the hair follicles. The only issue here is that the hair follicles only represent 1% approximately of the surface of the skin. Nevertheless, they could be interesting for treating acne, for instance, or other infections into the hair follicles themselves. So here we explore different sizes and different functionalities. We encapsulated a molecule and we also labeled the nanogels, following the same approach that Michael followed in the former study, but in this case by Fitsum. And what Fitsum could find here, I just take you to the very last part here on this figure, is whenever we have a higher sizes, these are accounting for nanogels with diameters around 500 nanometers. Whenever we compare this versus diameters of 400 or 100 nanometers, we could see that the 500 nanometers penetrate very deep as compared to the 400 ones, and as compared to the 100 nanometer ones that are not included here because they didn't penetrate into the hair follicles. And what we could see is that the loaded molecule penetrated a bit deeper. So this was working as a delivery system. And at the moment that we increase the temperature and we cross above the transition temperature, which are these lines here, so we have 35 versus 37, we can see that whenever we increase the temperature above the transition temperature, the loaded molecule can penetrate a bit deeper. That was saying about the mechanisms of penetration into the hair follicles. Just to finalize this section, I would like just to focus onto the delivery of therapeutic cargoes. We tried different molecules, from very small ones like dexamethasone, doxorubicin, and small peptides, of 6 kilovolt molecular weight. But as well, we tried very big proteins, or big compared at least with dexamethasone. And what we could see here is a threshold of responsive modalities for our materials. So the bigger cargo molecules were able to be retained inside of the nanogrels for longer time, and we only release slowly or faster depending on the properties of the materials whenever we cross the transition temperature. Today I will focus on this molecule here, etanercept. There is anti-TNF alpha molecule. It's a fusion protein, and for us it was very interesting because this is a molecule that is commonly used in the cases of severe psoriasis when the patient need to decrease the inflammation, and these are typically injected. So this is a systemic application for a disease on the skin, which doesn't make much sense. But since the molecule is too big, does not penetrate through the different stratum corneum layers by itself. We propose here to encapsulate these proteins within our nanogels. We run again the same approach of nanoprecipitation, very similar to the work of Gregor I showed you before. But in this case, as we count with the thermoresponsive nanogel system, what we did is to run a precipitation water in water, cold water that it works as a solvent for the polymers, and warm water that it works as a non-solvent for the polymers. This is what we have here. So PBS at 4 degrees and PBS at 37 degrees. And whenever we injected these molecules inside of the water, they were able to precipitate, form nanoparticles, and these were able also to encapsulate etanercept either post-synthetically or pre-synthetically. The studies that we did by zeta potential to analyze the surface charge of the materials enable us to see that etanercept has a strong negative charge, but at the moment this is encapsulated within the nanogels, this negative charge is hindered. This is giving us a hint that the protein is actually sitting within the structure of the nanogel and not on the surface. And then whenever we study the penetration of this protein into reconstruct the human skin, this is a study we did with Sarah Hedrich in Berlin. We could demonstrate that the protein, of course, whenever it's formulated only with buffer, is not able to penetrate the Stratum Corneum, but at the moment we formulate inside of our nanogels, this penetrate deeper, even up to the dermis. We could demonstrate the anti-proliferative, anti-inflammatory effects. We didn't see any sort of toxicity, which is a common issue on penetration of that one can see on penetrations of proteins. And then when we also analyze this anti-proliferative, anti-inflammatory effects, we could see that at the moment we formulate and we apply the encapsulated etanercept, we could recover most of the biological indicators to the normal state. Here is the production of TSLP. That is a protein that typically picks up at the moment we have an inflammation on the tissue. This was also published in Theragnostic in the cover, and this was published in the 2 years ago, 2017. Just to give you an idea of the other approaches that we follow in the group, and this is a paper that we haven't yet published, and this was a development that we did with 2 of our group members, now doctors. Joy Carr, he's actually from Pune, now he's doing a postdoc in Dresden, and also Lorin Toine, that she finished the PhD in my group last year. And here what we did is just use a very similar approach for the synthesis of the nanogels, but we provided these nanogels with disulfide linkages that it could be cleaved by the glutathione activity in the mucoses. What we propose is that the materials will degrade inside of the mucose. At the moment they get in contact with mucose, and then we'll expose thiol groups. These thiol groups should be able to interact with the thiol groups of the mucose, and also the smaller fragments should be able to penetrate deeper on the mucose. And well, we study, of course, the degradation by GPC and DLS, both in contact of glutathione and mucose. But more importantly, we continue this study with the group of Sarah Hedrich. Now she's in the University of British Columbia in Canada. And in this case, we encapsulate it and follow the penetration of etanercept. And this is what you can see here in green. So etanercept, when it's formulated in buffer, does not penetrate the mucose. This is a lung model that she has developed in her group. And here we can see this is basically staying on the upper part of the mucose, does not penetrate much. At the moment we formulate etanercept within the non-degradable nanogels. There is almost no penetration of the etanercept on the mucose, but at the moment we have the protein inside of the degradable nanogels. We have a stronger penetration of the etanercept into the deeper layers of these mucosal models that she developed. And of course, we continue working with these materials, and now to have even smarter materials where we can encapsulate hydrophobic moieties and different modalities within hydrophobic pockets in our nanogels. We have one of our postdocs, Neha Tiwari, I mentioned her before. She's originally from Pune, from NCL, and she's working in my group almost since 2 years, developing smart nanogels where we will try to include all the knowledge that we have obtained over the years. So just to come to the conclusion of this, we were able to synthesize different materials to demonstrate their potential and to understand mechanisms and penetration of the nanogel under cargos. And now we are also exploring mucose-degraded nanogels. We are exploring now here in San-Sebastain how to use the knowledge we got on different applications and ophthalmology against cystic fibrosis and wound infections and mucosal vaccinations. And we got funding from different agencies here in San Sebastian and in the local context, regional context, and also in the national context. But well, just this will be a story to tell you in a couple of years. Hopefully we will get nice results about that. I come to the acknowledgements. I have to acknowledge, of course, several of the group members that did this work and the current group members that are working on these new approaches. Of course, the funding agencies and all of you for your attention. So now we discuss the questions.
Thank you, Marcelo, for your wonderful presentation. We had more than 100 attendees. We all would have liked to actually come and meet you at your Polymat Institute, but this interface of internet, through internet, we have been able to actually be at our homes or offices, and attend this webinar offline through this wonderful interface bought by the internet. So how much time you have, because you have some other assignment after 3 to 4 minutes?
Yeah, I would say, yeah. Let's say 5 to 10 minutes. Yeah. Okay.
So before, I think you read a few of the questions in a chat, which I have recorded. If you don't want, I have first very light question for you. So which of these materials which you have been actually designing, synthesizing, characterizing, spending a lot of money, funds, right, collaborations, are most exciting to you? Which of either the conjugates or nanogels or the recent one which we actually made thermosensitive, which one you excite, which you want to take it for long term?
Well, I think exciting is everything we do. I really like the work we do in our lab, but of course it excites me a lot whenever we get closer to actual application. This was not the focus of our group in the last 10 years. So the focus, as you could see here in the presentation, it was rather scientific, trying to understand the interactions between the materials, the response they have, and the tissue. Yeah, and of course, since I have a scientific background, everything that excites me the most are the most complicated things. Yeah, I really like the nanogel with a different multi-stage system. But I also understand that at the moment that we have to come to apply this in a technology, we need to think on simplified systems. Yeah, so the super excited systems, of course, are the complicated ones that we developed. Yeah, and I think we will carry on exploring the applications, let's say the knowledge we can gain from them when it comes to interaction with the biological interface. But then I will also step back, and the excitement in some of the cases, try to move into simpler systems, and also try to explore now in the next 5 to 10 years in our group the actual applications and the translation into a technology. Yeah, so these are some of the new things that we show there. And also at the very end, I show you that we are also liking now excited to explore ophthalmology, for instance, as a field of application. But of course, there we will have to look into, let's say, from the engineering point of view, less exciting systems, but of course, more feasible from the technological point of view.
I was expecting you would say that the work which we do combiningly on CTC would be more exciting. Thank you. So
I didn't say that because I didn't show it. I didn't show it now. So
all the attendees, in the interest of Dr. Marcelo's time he has to run, I will actually ask questions on your behalf. So one of my colleague, Professor Chabisquar from MITWPU, asked a question actually, which of the polymers are most suitable for a conjugate?
In the general point of view, or the ones I presented now?
So it's a very general question. So maybe chemistry or size.
I think number one, I think the point is whenever we come to conjugation, we have to consider simple, at least simple chemical pathways to get into a conjugatable polymer. Yeah, so we have to activate the polymers. But it was also, of course, very important to have an inert platform. Yeah, so an inert platform that per se will not specifically interact with tissues or protein. This is why we were excited by polylysarol, because polylysarol is antifouling, will not interact with proteins, with cells, viruses, etc. by themselves. So it could be a very interesting platform. And the hydroxyl groups are very easy to modify. Yes, okay, modifying and pinpoint modifying the structure and polyglycerol, we can decide whether we want to modify an hydroxyl group on the exterior, on the intermediate levels, or very much in the core. And this was a very interesting molecule. What I will say is the limitation of these dendritic molecules is, of course, they're spherical. They have plenty of functional groups that are very interesting. But on the other side, they lose solubility very quickly. Yeah, so linear polymers on that regard could be very interesting because they will be able to self-assemble in a way to keep the solubility at least to a certain extent. No, not as a pristine molecule with very small size in solution. They will typically aggregate and get deeper and bigger sizes, moving from a polymer typically with sizes of 5 nanometers. They will probably couple an hydrophobic drug and will self-assemble into an aggregate of 100 nanometers at least. Yeah. But if I will have to choose, I will say this will depend, of course, a lot into the drug we want to use and the properties the drug have. But I will play between the two materials. Yeah, having the dendritic system for certain applications, linear ones for another applications. Of course, keeping always in mind this platform idea I just discussed. It's a very general answer because the question is still general. No?
So there are our student Vikram, who is now doing his master's in what is nanogels in temperatures like Indian temperatures very high. And then we have a professor outside from Srinagar Institute. She calls about protease enzyme studies. How could we do, etc. These are the two questions.
Okay.
So stability of nanogels.
Yeah. So what I can say is, of course, the thermal stability of the material by itself, it will depend on the thermal stability of the building blocks. Yeah. I'm just answering about thermal degradation, right? This will not vary a lot. What it could vary a lot is the thermal stability when we come to properties in solution. Yeah, so NIPAM, for instance, which is the golden standard in thermoresponsive materials, is typically readily soluble below the transition temperature as a linear polymer or as a branch polymers. But whenever we cross the transition temperature, this material precipitates. Yeah. So whenever we would like to have the NIPAM still stable at above the transition temperature, we will have to consider to copolymerize it. In our case, the copolymerization was with this cross-linker that is polyglycerol and is highly water soluble, and therefore the huge amount of polyglycerols we have there, it has a weight composition of 30%. It enables the nanogels to be still stable above the transition temperature. So you have to play, of course, enhancing the solubility of the materials above the transition, otherwise you will have them precipitated. This, in our case, was desired for systemic applications. I didn't show the systemic application we did with these nanogels. So for topical drug delivery, this wouldn't be an issue. But whenever you would like to target systemic application and you would like to have the polymer still circulating after releasing the drug because you would like to have excretion of the polymer, then you have to consider, of course, stabilize it above the transition temperature.
So my question here,
there was another question. There was another question, right?
Oh, so how do you actually estimate, say, protease activity in vitro? Okay. Do you do that, or
how we study? You mean?
Yes, protease enzymes effect.
So this will be a typical degradation study. Yeah, so you can do it very simply by just checking the size of the nanoparticle in solution and then incubate it with these proteases at different concentration, different times. You can very simply follow the DLS peak, if considering you don't have aggregation when you aggregate the enzymes and so on and so forth, right? You can follow very simply by DLS. A bit more sophisticated and more precise will be to follow the fragments by GPC, gel permeation chromatography. And a bit more sophisticated is what we did in Gregor's case, where we have this Fred pair. At the moment, the Fred pair is separated from each other. We recover the fluorescence of one of these dyes. And then there you can also track the fluorescence recovery of the material upon degradation with the proteases. And the very common study you can do with proteases, starting with the trypsin. Yeah, trypsin is a very general protease that it will cleave almost everything. And then whenever you want to get into more a more sophisticated system, then you can start testing certain enzymes. Yeah, either proteases, MMPs, esterases, so on and so forth. I just mentioned a couple of techniques, but of course you can try many others. No, you can check viscosity of the media, you can check rheology properties, gel-like properties of the material, etc., etc. Yeah.
So overall, what is the status of nanomedicine? So Umesh asked, so this might be a last second question. So where have nanomedicines, nanomaterials are actually reached in therapeutics?
Well, this is a challenging question. Yeah, I think what we all see, or at least many people saw nanomedicine at the very beginning with a strong optimism. Yeah, but not so much realism. And we know that nanomedicine still is a field on at least from the technological point of view is still growing. Yeah, I think from the scientific point of view, I feel more and more that we are already reaching a limit. Yeah, so nanomedicine, especially when we consider it from the point of view of a polymer chemist, nanomedicine is out there since several decades. And since several decades, there are thousands of research groups synthesizing thousands of different nanoparticles. And probably we are already reaching the limit of the kind of nanoparticles we can synthesize. And probably this is why I also feel that the focus should start moving slowly, at least into looking into the technological development of these materials. I think we're still on the infancy when it comes to the therapeutics, for sure, because we know that the therapeutic development takes at least 10 years, yeah, and probably $1 billion. But of course, I will say something in pro of the CTC technology. I didn't mention it today because, of course, I didn't talk about CTC, but we have several studies done together with Jayant, and of course, we are super excited about that. And when it comes into moving nanomedicine into more nanotherapeutics, sorry, nanodiagnosis, I would say we are much closer to the reality in that case, because now the regulations are lower, the path into the commercialization is shorter, and therefore we need a shorter time and less funds to complete the development. And of course, the CTC technology that Encore Discover developed is of course a proof of that. No, and also I didn't get into the details of this vaccination that Moderna now is moving into the clinical trials after positive results with 8 patients in the US against coronavirus. But what I heard is there is a lot of nanomedicine involved there behind, yeah, the transport of these RNA fragments. As I said, I didn't get into details, so I cannot say much, but I will say that there is potential still. Of course, there is still a lot of potential, but we have to be realistic about this issue. Thinking on therapeutics will need at least 10 years and $1 billion. This is nothing we should forget about.
So, very difficult question. When are we going to meet, or when are you going to travel to India?
This is a very difficult question. I really don't know. No, so we cancel all the trips we have planned this year, and I hope we can travel next year again. Yeah, but it will depend a lot into development of vaccination against coronavirus, or perhaps a lucky mutation of the virus that will be much less, will decrease the virulence of corona. But I hope next year. But I think nobody here is on place of promising anything for this year, at least, no.
So before I say thank you and goodbye, I think we have team from your Polymat Institute like David, Neha, probably Maria. We have company people, Abhijit, Sanjay. We have academicians. We have SPP University, Atul. We have our own company people, actuaries, and most important, Balram and Smriti, who worked with you. They are also here today. And on behalf of my institute and dean, Dr. Kuchekar, and we are now MIT World Peace University School of Pharmacy since last 2 years. And so many PhD students, master students, graduate students, I think 10 institutes today we see in this 100 numbers that has got a very wonderful response. I see some professors from other allied biotech specialists like Preeti. I see Medical Institute scientist Yuvraj and Shaswat, which you know. And for a short time, I think I must acknowledge everybody for their time today to listening to you. And it has been pleasure, Marcelo. And I say goodbye.
Let me say thank you to all of you that took your time today out of your families and out of your common activities. So thank you very much, and congratulations then for the new nomination on the university. Congratulations to the authorities, and of course my warmest greeting to everybody from my group. Of course, that I see some names here on the audience, and current group, I mean, and also to the former collaboration links we had. No, I'm very happy to hear that you have some of our former group members there and some of our former colleagues. So in case you have more questions, of course, you can feel free to send me questions, and if you want to send me the feed from the chat here, because I saw there were some questions popping up from now and then, just send me and I will email you answers if I have some.
I will communicate to them your email ID.
Perfect. Thank you very much to everybody then, and I wish you a nice afternoon now here, evening.
Take care, and you have a long day ahead. All the best. All the best, and
Same to you.
pass on.
Greetings to your families.
Thank you. Goodbye.
Bye-bye.
Hello. Hello.